So today we're going to continue with the discussion of meiosis and then move on to showing how you can apply a lot of these principles we've been teaching you to various problems, and the one we'll tackle today specifically is the concept of DNA testing. Okay, so we're just going to continue then, finishing chapter 13, and the book has a little bit on DNA testing, but I'll go into it in quite a bit more detail here, I think, today. Okay, so we left off yesterday, or last lecture, with talking about meiosis, and I just want to clarify something. So I think I created some confusion by saying that I quibbled with this definition of this end of meiosis 1 is haploid, but by the strict definition of haploid diploid in terms of chromosomes, that's correct, because it doesn't have two copies, the two homologs, right? It has two DNA copies of a single homolog. So in that case, it is haploid. And those of you who are taking the lab, you've had a lot more explanation of this. But what might also be useful to keep in mind is this C value, so the amount of DNA that's actually there in the cell. And so, for example, as the cell in mitosis goes through S phase, we still consider it diploid the whole time, but in fact, actually, it's going from 2C to 4C at that point, right? And then in meiosis, then, when you have this first meiotic division, you have this separation of the homologous chromosomes, and so you're at 2C now. But by that definition of what's going on with the chromosomes, you would consider it haploid. So if you keep both things in mind, it helps you to understand what's going on in meiosis. And it's very easy to understand that all the way at the end of meiosis 2, it's haploid and 1C. So there's just a equivalent copy of one chromosome there in each cell. It was interesting, though, because I took a poll of 7 MCB faculty as to who would call this haploid, and none of them called them haploid at that point, which I think reflects the fact that right nowadays we focus so much on DNA and genes and not so much on chromosomes. But if you think about chromosomes, that is then considered haploid for the reasons of the strict definition. Okay, so moving on from there then. And I should mention that even one of the MCB faculty members I asked was the person who actually discovered cohesin. So works a lot with chromosome segregation, and he was still uncomfortable calling it haploid. Okay. Yeah, just as it's shown up there. Yep. Okay. And the reality is, of course, because of crossovers, you could actually have the two different alleles that had come from mother and father. Okay. But that's not true for the entire chromosome. Okay, so then we talked about how you get random segregation, and one thing was that when you have multiple chromosomes, it's easy to understand how the different chromosomes can segregate randomly because they can line up in different arrangements at metaphase and then get separated in such a way that gametes can have a random assortment of those different chromosomes. Okay, but in terms of then if a gene or two genes were on the same chromosome near each other, how would you end up getting random assortment? And we talked about how Mendel was in some sense lucky that he picked alleles that either were on separate chromosomes or far enough apart on the chromosomes that they always segregated separately. So some people say, well, he must have thrown away some data. He must have observed something that didn't follow his pattern. If he did, he was still quite a wise man for understanding the pattern in the data he had, and he probably just didn't understand the other things that were going on. Anyway, so by this process then of the recombination that occurs, you can get all the combinations possible. Okay, but the frequency of the segregation for genes that are close together on the chromosome is proportional to how close together they are. So if they're very close, they're unlikely to separate. And as they get further and further apart, the probability goes higher and higher. And so, in fact, usually chromosome arms are so long that genes at opposite ends of the chromosome arm segregate as though it was random. Okay, so, and then we highlighted the differences between mitosis and meiosis, and there's three very unique things about meiosis, and they all occur during meiosis 1, and they're listed here. And again, we went over this last time. So synapsis and crossing over in prophase 1, so homologous chromosomes physically connect and exchange genetic information, right? So that's what's going on right here. And I think a student asked one of the GSIs whether the way this is drawn of how the pairs are lined up to each other is meaningful. It's not, because in fact, they're not actually condensed chromosomes when this recombination occurs. They're actually unwound, and the DNA is undergoing this recombination process, not as condensed chromosomes. Okay? So their orientation to each other is actually fairly random, but nevertheless, the homologs are lined up, right? The strands at least. Okay. So, and then at the metaphase plate, you have pairing of the homologous chromosomes instead of just individual replicated chromosomes. So again, that's shown in the difference right there, where the homologs are lined up together, so you get what we call tetrads, as opposed to this sort of configuration of sister chromatids. Okay. And then finally, at anaphase 1, it's the homologous chromosomes instead of sister chromatids that separate. Okay. And then some of the things that are going on, sister chromatid cohesion allows sister chromatids of a single chromosome to stay together through meiosis 1. Okay, and that's different than what you think about in mitosis. And there, again, these protein complexes called cohesins are responsible for this cohesion. They're the same things that were acting during mitosis. But in mitosis, those cohesins are cleaved at the end of metaphase, and then the sister chromatids can separate. But in meiosis, the cohesins are cleaved along the chromosome arms in anaphase 1, right, which allows the separation of homologs. But the centromeres stay connected. Okay, and the cohesins don't cleave there until anaphase 2, which allows separation of sister chromatids. And that's why you get, that's responsible for the pattern that you see or the events that go on in meiosis. Okay, and again, that's just a summary diagram giving you the similarities and differences. Okay, but the idea is that in mitosis, you go from a somatic cell with this 2N complement of chromosomes to two cells with 2N. Okay? But in meiosis, you're making the gametes, and you end up going from that same sort of cell, but then you end up with the four products of meiosis are each haploid, okay, and only contain a single copy of any given gene now. Okay? And so that means that they have an allele either from the original mother or the original father of that person. Okay, and then the crossing over involves these chiasma that we talked about, and that allows recombination. Yeah. It becomes connected, not in the middle, but it becomes connected by the arms. And we'll talk about that, because one of the things that has to happen is that when the microtubules pull on it, there has to be tension. And so without the tetrads being connected by that recombination event, there would be no tension. We'll get to that, but that's how they're connected actually is by the recombination. Okay. Yeah, because the way it's drawn here, you don't see them connected at that point, right, the homologs. But what connects them is that recombination that's occurred. No, so that's not between the homologs. Okay, so then I left you with this question about mapping, or we discussed mapping. And so we talked about if you had genes on the same chromosome that were relatively close together, what would happen? And this is the typical kind of test cross scenario. So what you do is you make the heterozygote, okay? And let's pretend this is the configuration then of the chromosomes in this heterozygote. And so the two genes we're talking about are rosy and frizzled, and these affect the eye and pairs. And so let's say this chromosome has rosy minus, frizzled minus, and this one is rosy plus, frizzled plus, okay? So that's the heterozygote. But if you then cross this heterozygote to fly that's homozygous for the recessive alleles of both genes. And this is called a test cross because it makes it very easy to see what's going on because this chromosome is giving all recessive alleles. And so it makes it easy to follow what's happening on this side. Okay? And so these are the different possibilities that you have. Okay? And so these are the phenotypes that you get. And again, if you think about this, making this test cross in this way makes it very easy to see the 4 different phenotypes that you get, okay, and relate them to genotypes. So what you can see, though, is that if these were segregating randomly, you would expect a 1-to-1 to 1-to-1 ratio. Okay? But it's skewed. You don't get that. So 2 are very abundant and 2 are relatively rare in comparison. And so then if you look at this and you think about the original orientation of the chromosomes, you see that the parental orientation, so having the 2 rosy minus, flux minus, having those 2 together, or having the plus and plus together, are the ones that are much more abundant. Whereas the recombinants, right, that would occur if you had crossing over, okay, those are relatively rare. And so that's a measure of the recombination frequency, okay? And so you can make a calculation then. You can take the sum total of the flies that you've got, and you can divide the recombinants by that total, and that gives you recombination frequency. So in this case, it's about 10% recombination frequency. And Sturdivant came up with a nomenclature of Polynos math units, okay? And then we discussed that for 2 genes on different chromosomes, or 2 genes that are very far apart, so there's basically always a recombination event separating them, that number would be 50, the recombination frequency, okay? And the smaller that recombination frequency, the closer those 2 genes must be on the chromosome. So then I left you with this question. So we had another example here where we talked about if you tried to collect these numbers, okay? So again, rosy is a recessive gene. The homozygous gene has rosy-colored eyes. Frizzled has these bent hairs. Again, it's a recessive mutation, okay? And we said that, so you did this experiment. You were basically trying to do the same sort of back cross or test cross experiment. So you took a fly that was heterozygous for both rosy and frizzled, and you mated it to a male, in this case, that's homozygous for both rosy and frizzled, okay? So a heterozygous with a homozygous. And you got, these numbers, okay? But then we said, in quotes, your friend, right, did the same thing, okay? And they got a very different set of numbers, and we'll put them both up there on the screen, okay? So you think you did the same experiment, you got very different numbers, okay? And I left you with that question at the end of last time, why, okay? So that's what I want you to answer now, is why do you get 2 very different looking results, okay? So here are the possibilities that I want you to entertain, okay? Okay, what I'm going to do is give you like 1 minute, okay? Write down your answer on a piece of paper because I'm going to put up the iClicker thing. The question's going to go away for a second, and then vote, and then we'll go back, okay? Yeah, pick the best answer. Okay, so I'm going to let you vote. Okay, just a second. Okay, I'm not going to put it up on the screen, but I'm going to give you 30 seconds to vote. So you can start voting now. Okay, just a few more seconds. Okay, I'm going to stop it now, okay? Okay. All right, so let's see what we got here. Okay, so most of you voted for B, okay? All right, so now let's go back here. Okay, so most of you said B, okay? And that's that it depends on the genotypes of the flies used to create the flies heterozygous for both rosy and frizzled. And the idea is that you and your friend made them in different ways, okay? And so the idea there is that the actual relative positioning of the alleles on the chromosomes is different depending on how you went about making your heterozygous fly. And that's sort of illustrated here, that there are 2 different ways you could have done this. You're making an animal that's heterozygous at both alleles, but you can see that there's 2 different ways you could have generated that same phenotype. But the nice thing is if you, of course, write them out as chromosomes, you realize that these are not the same. So they, in fact, are heterozygous at both for both genes, but this one has the rosy minus and frizzled minus on the same chromosome, and the pluses and pluses, and this one is not that way. It's plus and minus and minus and plus. And so that means, and you might notice, I think I turned around the way I labeled this, but it doesn't matter because I didn't tell you what is maternal or what's from mom or dad. But that's the idea, is that those chromosomes have different orientations like that. And so then that's why you get these different numbers, okay, because of that. And so you should make sure you understand that, okay, because it's important when you do a test cross that you really understand the orientation of the genes going into the test cross. Otherwise, you're not really going to get the definition of the recombinant and the non-recombinant chromosomes correct. All right. So, so if this is the case, right, and these are the numbers I got then, okay, why don't you go ahead and calculate the map distance? We won't vote on this. Just go ahead and calculate it. You don't have to get a calculator out and do it all the way to the percentage. Just write out the numbers that will give you the right answer, okay? You can just do it for one or the other. You don't have to do it for both sets of numbers, okay? But you should understand how you would do it for either set of numbers. Okay, and this should give you an answer that's a percentage, right, a fraction, and then you turn it into a percentage. Okay, so hopefully then if you calculate it, you get the map distance, and you've got something like this. So on this side, these would be your recombinant chromosomes, right? And on this side, it's these 2 that are your recombinant chromosomes. And you should be able to see that from the way that these chromosomes are arranged. So in this case, having the heterozygous, the minus minus or the plus plus together, right, are the parental orientation, whereas that's the opposite here. And so that's why these numbers are different. But then when you go through the calculations, you get the same answer, which is what you should be getting, right? Because it doesn't matter what the arrangement of those alleles was. It's the distance between those 2 genes on the chromosome that counts. Okay, so you can use that kind of information to create a genetic map of an organism. And this was a classic traditional way of actually looking at chromosomes, mapping genes, doing lots of genetics. And so you can see here, for example, 3 genes in flies that are mapped. One of the things you may notice is that the sum of these 2 is larger than this. Okay? Does anyone know why that is? Why that would happen? Any guesses? What's that? You don't count what? It's that you, yeah, in a sense, and it's because of, you can actually get 2 recombination events, okay, in the interval. So the further apart you get, the more likely it is that every now and then you'll get 2 recombination events in the interval. So that's illustrated here, and that'll throw off your calculation. So what you're assuming is you just get these single recombination events, right? But you can get 2 recombination events, and you have to take that into account whenever you're doing calculations. If you just have 2 genes that you're mapping, you can't really see the frequency of the double recombinants because they would look like the parental chromosomes. So that's right, you're overcounting that family then, right? That class. And so the more genes you have and the closer together they are, the more accurate a map you can make, because the real accurate map is to add up all those individual distances instead. Okay, so again, and repeating what we said too, that when genes are far apart, that number hits 50. So one of the weird things that you can get is you can make a genetic map of a chromosome that's, say, 120 map units long. Okay, and that doesn't make sense for a minute, right? Because that means that genes at the opposite ends of those chromosome maps are going to act as though they're on separate chromosomes. They're so far apart. And you only get that number because you've summed up lots of little intervals. And so then you can tell that 2 genes are linked, even if they're opposite ends of the chromosome, and seem unlinked by a simple cross of those, because you've linked them with all these intermediate genes. Okay. And then, let's see, so Sturtevant used these recombination frequencies to make a linkage map for fruit flies, for example. And then, if you've ever seen pictures of a chromosome in flies from the salivary gland, they're what we call polyT, and there's actually many, many copies all stuck together. So they have a banding pattern that you can actually see pretty easily in a microscope. And people eventually realized there was a correlation between those bands and gene positions, and so you could physically relate that to the chromosome. And that led to these cytogenetic maps that were used a lot to then help look at breakpoints in chromosomes where those genes were and so on. And you can create these kind of maps. And they were actually extremely useful until recently, when it became so much easier to genotype the DNA instead and just sequence it and figure out where you were. Okay, so here's your next question. You don't have to try to answer it on your clicker. Okay, so how many do we have that go for A and B in the room? A few. Okay. And I assume the rest of you are a combination of C, D, and E together. Okay, but my point in this is just that, so we show you a lot of fly genetics and P genetics and so on, right? And maybe you're like, what's the point? We try to show you a little human pedigree, right, to illustrate that these things still apply. But what I thought I'd do for the rest of today is try to give you a more solid example of how you can take the information you have and start thinking about it in terms of human genetics, and give you something that probably you have some familiarity with, you've heard about, but maybe don't know exactly how it's done. So I'm going to end this for a second. Give me 1 minute here. Okay. I'm going to try to make this work. Okay. All right. I'm going to play the whole thing. I'm going to play 2 parts. Hopefully the sound is working. Okay, how many of you watched the Maury Povich show? All right. Okay. So let me turn the volume up.
When she first met a guy named Wayne, they immediately hit it off, quickly fell in love, and when Karen found out she was pregnant, they were both excited. So much so they got engaged. They set a wedding date. They prepared to be a family. Unfortunately, all of Karen's plans came crashing down when she was 4 months pregnant and found out she was having a baby girl. And this is why I was smiling. Because when she found out she was having a girl, that's when Wayne started to completely deny he was the father of a child and completely abandoned her. And so here, into this world comes little Lorene. And Karen, he says he's not the father because he says he doesn't make girls.
Okay, I'm going to skip forward. Go through all the yelling and everything. Okay.
It's like him. Okay. When it comes to 2-month-old Lorene, Wayne, you are the father.
Okay, enough of that tragedy. Okay, so the question now is, what's on that piece of paper that he's reading? Okay, so how does he know that that guy's the father? Okay, so what goes on, right? So probably in the old, not that far back, there was very few ways to tell. I mean, the first one is what she said at the beginning, the baby looks like him. Okay, not quite too accurate. I mean, to a rough approximation, right? Makes people suspicious, or but as we talked about, lots of physical traits of humans are polygenic, and they're affected by environments and things like that, right? So the idea is to use genetic markers, and in a sense, that's what you're doing when you look at 2 people and you compare them, right? You're thinking about the effect of heredity in genes. But what you really need to do if you were going to use some genes is to be certain of the pedigree. So, if there are 2 people who have blonde hair and they have a kid who's a brunette, well, if in both families there's plenty of instances of both blondes and brunettes, it makes some sense, right? And you can think about combinations of recessive and dominant alleles and the multigenic trait and so on, okay? And anything you do like that can be confounded by epistasis, interactions of genes, and by environmental effects, okay? So, like how tall a child is relative to his parents. Well, it also depends what the nutritional level of the parents was relative to the child, and so on. So there's a lot of confounding factors. So instead, what we use nowadays are DNA sequences, okay? And we use differences called polymorphisms between equivalent homologous regions of chromosomes, right? So you can think of that in a simple way. So imagine that you have 2 DNA sequences, okay? And so these would be, say, from the 2 different copies on a gene in a somatic cell. So can anyone see the difference? Okay, no, probably not. Okay, so there it is, okay? So there's a single nucleotide that's different, okay? But that's an informative trait, okay? And that's a polymorphism. And this could actually be a causative mutation that gives a phenotype. So, for example, in a fly, this could be a mutation in the ROSY gene. And we'll talk more about the nature of mutations later and the idea of how you make protein and so on. But this could be, we talked about how, alleles were the same gene, they just were a slightly different DNA sequence usually that had a phenotypic effect. And so it could be that this is the wild type gene that encodes a normal enzyme, and that change of a G to a C makes this so that it doesn't make the proper enzyme, right? And that would be the idea of a ROSY plus, ROSY minus. Okay? So, but in fact, the human genome is filled with differences like this that don't actually have a visible phenotype. Okay? So they just are what we call neutral mutations, right? And there are lots of them in the genome. And the reason that they're there is we talked about DNA polymerase is very accurate, right? But it's not 100% accurate, right? It makes a mistake 1 in every 10 million base pairs. But if you think about the amount of DNA you have and how many people are there, okay, that means that mutations are going to accumulate. And then we won't go through the sort of population genetic models, but some of the mutations can go to fixation, and others are just at varying frequencies in the population, right? At any one moment. And you remember your Hardy-Weinberg equilibrium.